Hello students, today we will study about the enzymes. What are the enzymes? Enzymes are the biological catalyst. A catalyst is a substance that speeds up a chemical reaction. For example, A is reacting with B to produce C. In the absence of catalyst, the reaction will take place at a longer interval. When the catalyst is added, the rate of the chemical reaction speeds up. For example, in the Haber's process, nitrogen reacts with hydrogen to form ammonia. But when the catalyst is added, the formation of ammonia speeds up. The catalyst used in this case is nickel. So it is also a metal catalyst. In the case of a chemical reaction that is occurring in a living body, for example the metabolic reactions, the catalyst is termed as enzyme. So the reaction that is occurring within a living body or living cell, its reaction is speeded up by the addition of the enzyme. So what are the characteristics of enzyme? Number one, the enzyme speeds up a chemical reaction. Number two, it is not utilized in the chemical reaction. It means the enzyme is reproduced at the end of the chemical reaction. It just speeds up, but it is not utilized in the chemical reaction. Chemically, the enzyme is protein in nature. Sometimes it can be RNA, but in most cases, the enzyme is protein in nature and it is very specific in its activity. For example, the only nickel catalyst is used in this Haber's process. Same like in the living system, a specific enzyme will be used for the specific chemical reaction. For example, if we are using RNases, they break the RNA. If we use DNases, they break the DNA. So the enzymes are very specific in nature. They depict the reaction. For example, in the case of an enzyme catalyzed reaction, the reactants are known as substrate. So this shaded area is the substrate and this is the structure of an enzyme. Enzymes are mostly globular in structure and they have the active site. Active site is the region where the substrate binds. The substance that is to be converted into the product. So the substrate is binding with the enzyme, and with a reversible reaction, the substrate and enzymes, they form a transition state or an intermediate state that is known as enzyme substrate complex. So this is the enzyme and the substrate complex. We write it as ES, enzyme substrate complex. Further, when the reaction is proceeded, the enzyme is released again as it was utilized in the chemical reaction, and the substrate is converted into the product. So we can say that the enzyme just play the role in speeding up the chemical reaction and it is not utilized in the chemical reaction. So the activity of enzyme can be illustrated by this energy diagram. This diagram shows the process, the reaction that is proceeding in this direction, and the energy utilized by the substrate to be converted into the product. We know that the substance needs, or the reactant needs, the activation energy. What is the activation energy? Activation energy is the minimum amount of energy that is used for the chemical reaction to proceed. So if the reaction is uncatalyzed, this one, the reaction, I can label it as A. In the A, the reaction is not aided by the addition of enzyme or any catalyst, but in the B, the reaction is aided by the enzyme. So what is the difference? In reaction A, the amount of energy or the amount of activation energy is higher, but with the enzyme, the substrate need lower activation energy. So this is a difference in the activation energy. So we can say that enzymes actually lower the activation energy, so the reaction takes less time to be proceeded and converted into the product. The activity of enzyme is illustrated by the two models, the log and the key model and the induced fit model. Log and the key model says that just like a specific log is opened by a specific key, the specific substrate, the shape of the substrate should be complementary to the active site of the enzyme. I just mentioned that the enzyme is globular in structure, it is protein in nature, but the most important site over here is the active site. It is the site where the substrate binds and it is converted into the product. So how the substrate binds in the enzyme, that is illustrated by these two models. The log and key model says that the active site of the enzyme is rigid, just like the shape of the key and the enzyme. They should be complementary. For example, this shape is adjustable over here and this shape is adjustable over here. the shape of substrate should be like the active site of the enzyme. Otherwise, the reaction will not proceed. But the induced fit model says that the active site of the enzyme is not rigid, it is flexible. So this is the major difference. The lock and key model says that the active site is rigid, but the induced fit model says that the active site is flexible. So what happens when the active site is flexible? The shape of the substrate is not complementary with the shape of the active site of the enzyme, but when the substrate binds with the enzyme changes its shape and it adjusts the substrate into the active site. So actually, what is the role of the active site? It provides the space to the substrate where the both substrates react with each other and they are converted into the products. So as we know that the enzyme are protein in nature, the active site is also made up of specific amino acid. The amino acid sequence should be complementary with the substrates. Other than active site, there is another site in the enzyme in both cases that is known as the allosteric site. Allosteric site is the region other than active site where the inhibitor or activator can bind. For example, the substrate is binding with the active site, but an inhibitor binds over here. An inhibitor does not want this substrate to be converted into the product, so the reaction is stopped. The inhibitor stops the reaction and it does not allow the enzyme to work properly. When the activator binds over here, it allows the enzyme to work properly. So activator enhances the rate of the reaction and inhibitor stops the enzyme to work. So the allosteric site is other than the active site, but it also plays a role in the activity of the enzyme. Inhibitor or activator are the compounds that actually when binds to the allosteric site, they change the shape of the active site. So when the shape of the active site is changed, the enzyme is not able to bind the substrate. So we can say that enzyme is a biological catalyst and it is almost always a protein. It speeds up the rate of a specific chemical reaction in the cell. The enzyme is not destroyed during the chemical reaction and is used over and over. A cell contains thousands of different types of enzyme molecules, each specific to particular chemical reaction. An enzyme is a biological catalyst that is usually a protein but could be RNA, and the point of catalyst is to increase the speed with which a chemical reaction happens. And there are many enzymes that are encoded by the genome to make proteins or RNAs that speed up various chemical reactions to do thousands of different functions inside the cell.